Scientists often use measures of biological condition as a way of assessing ecosystem health because unlike a chemical measure, which measures condition at one point in time, biological measures allow us to integrate conditions over time and space. Now, another reason we use biological measures is because a lot of our regulatory programs are aimed at protecting the biology of the resource. And so by measuring biological indicators, we're getting a more direct assessment of how effective our management programs are being at protecting those resources of interest. Now, one of the big challenges associated with bioassessment is that it requires that we collect organisms like insects or fish and identify them individually under a microscope based on their physical characteristics. Now, obviously, this can be costly and time-intensive, and it can also be limited by the capability of our trained taxonomists, the availability of taxonomists, and how well our taxonomic keys are. So molecular methods provide a tool that we can apply to help overcome some of these challenges by not only reducing the cost and the time necessary to get our answers, but also by allowing us to improve the efficiency and the resolution and the accuracy of the data that we collect. Now, one such method that we're working with here at SCCWRP is called DNA barcoding. And essentially what DNA barcoding is it uses a small section of mitochondrial DNA that's well conserved from generation to generation, and we can use that as a genetic fingerprint or identifier of the organism of interest. Now, we don't expect that DNA barcoding and other molecular methods will completely replace traditional taxonomy, at least not over the short term, but it does provide another tool in the toolbox that we can use to improve the accuracy and the efficiency of our assessments. So SCCWRP scientists are working with experts in Canada, the US, and Europe, and our goal is to develop tools and approaches that will allow us to ultimately apply these new molecular methods for use in routine environmental bioassessment.